Okay, so I am discussing DNA replication, which begins obviously at the initiation of DNA replication. Okay, so the replication of DNA begins at specific sites along the DNA strand called origins of replication, which I drew in here and here. Initiation proteins at this point recognize a beginning sequence and separate to form a bubble, which I drew here. And at each end of this bubble is a Y-shaped area called a replication fork, which is basically just the point where the DNA strand separates, forming the bubble. The enzyme that helps do this is called helicase. And then after the helicase separates the DNA strand, single-strand binding proteins, which I drew in here and here, form along or join along the DNA strand to help keep it from rejoining after it goes through the helicase. And the unbinding of the strand, or the unwounding of the strand here, puts an added strain on the DNA strand over here, which sometimes causes it to twist tighter or break. So the enzyme topoisomerase binds to the DNA strand here to help alleviate some of that stress that could be caused or added to the DNA strand outside of the bubble. The two strands of DNA run in opposite directions, meaning that if we go from right to left, the top strand reads 3' to 5', while the bottom strand reads 5' to 3'. This is important in noting when replication because when replicating a DNA strand, the replication strand must run 5' to 3', which causes some implications down here in the lagging strand, which we'll get to later on in the video. So we're going to begin with the synthesis of the leading strand, which is often called the continuous synthesis because the top strand runs in a 3' to 5' direction. So the DNA replication of the synthesis of the next strand will run in that 5' to 3' direction that I just mentioned was mandatory. And in this strand, starting at the origin of replication, we have our RNA primer that's laid out, followed by our DNA nucleotides, which are assembled using the DNA polymerase 3, often shortened to be DNA Pol 3, and are often put into place or punched into place by this sliding clamp that clamps them down. Notice that we still have our helicase separating the strand before it, and our single-stranded binding proteins still bound to the DNA strand to help keep it from rebinding or rejoining with that lagging strand down here. So this, after the primer is laid, the DNA nucleotides keep going until the end of the strand, until basically it's all completed, which is one continuous synthesis of the next strand, which is why it's called the synthesis or the continuous synthesis. The lagging strand synthesis is a bit more complex. In this synthesis, because it runs in the 5' to 3' direction, the synthesis is going to be discontinuous and broken up into different parts to try to get it to run in that same direction that it was running up here. So we lay down our RNA primers here, here, and here. And it's just important to note that this one was forming towards the replication fork, and in the lagging strand, we're going to have our template form in the opposite direction or away from that replication fork. So we have our primase, which joins RNA nucleotides into the primer, which is here. We have a primase enzyme doing that. And this primer is then used to begin transcription for the lagging strand. When the primer is after the primer is made, DNA polymerase 3 or DNA Pol 3 adds to DNA nucleotides until it reaches the next RNA primer. And this segment of DNA nucleotides, which would be here if we had one more here completed, here and then here, are called Okazaki fragments. And the basic definition of an Okazaki fragment is just the DNA nucleotides between the RNA primers on the lagging strand. After the DNA nucleotides reach the RNA primer, this DNA Pol 3 detaches from the strand, or the Okazaki fragment, and this process continues throughout the whole synthesis of the strand. Then an enzyme called DNA polymerase 1, shortened DNA Pol 1, replaces these RNA primers or RNA nucleotides with DNA nucleotides. And these DNA nucleotides they're replaced in on the 3' end of the Okazaki fragment, which would be over here, because if we're thinking about that, there would be a 5' there and then a 3' there. And after the DNA polymerase 1 replaces the RNA nucleotides with the DNA nucleotides, an enzyme called ligase, which is shown here, forms a bond between the new nucleotides that was just laid down and the ones that we had put down during our Okazaki fragments. And the circle here kind of shows that binding of the two sections of the nucleotides. The enzyme ligase moves in that 5' to 3' direction, which we said at the beginning of the video is one of the requirements for the synthesis of DNA. And it's important to note that the leading strand and the lagging strand they both synthesize at the same time. So it's not like the leading strand synthesizes and then the lagging strand does, although the lagging strand does go a little slower than the leading strand because of the discontinuity between the RNA primers and the Okazaki fragments and that stuff. After discussing the process of DNA replication, we should probably take a step back to discuss why this is important and how does it affect us. DNA replication is overall efficient and fast. It produces about 40 nucleotides per second. It makes a mistake in about 1 in 100,000 nucleotides. It's important to also realize that if there is a mistake in DNA replication, that mistake will be carried on through the daughter DNA, and then that mistake will live throughout the DNA as it continues to be replicated. It will never be replicated as that former DNA strand. This has some benefits and some downfalls. If it's the mistake can be harmful and it could hurt our genes or like change our genes in a way that's not beneficial to our body. And other times it is beneficial. It can help us adapt to our changing environment, which is the idea of natural selection, that the strongest or the best will survive and that the weak will die off. So this definitely does have a double side effect, and it's one of the reasons why we should think of it as important, because DNA is our genetic material. It codes for all of our genes and our hair color and eye color. So although it's so small and abstract, people often think it's abstract, but it's got a much wider basis that we can really see in our everyday to day life.